Hello everyone, today we are covering gram stain preparation. We are studying bacteria, and in order to view the prokaryotes or bacteria, we need to do something called gram stain. We will prepare a microscopic slide, okay? And I have over here a culture, and this culture from inside, it contains bacteria. So I will grab some bacteria from here, I will apply it on the slide, and I will add it on my hot plate. This is my hot plate. Now I'm just using it before I open it. I'm just, I want you to understand what is it first. And this technique where I add a drop of water and the sample, bacteria sample, I will apply it on a hot plate until it's dried. The reason I do that is to fix the slide. We call it fixation. We need to adhere the sample on the slide, okay, for one reason. And the second reason is to make the organism harmless. We're not killing, we're just making it harmless. So once we dry the sample on the surface of the slide itself, afterwards we will start the technique of the staining. We will stain the bacteria, we will color it in order to view it under microscope. So just give me a minute and I will prepare it for you. So here's the drop of water, okay, and I will use this disposable stick. Usually we use it metal and we have to heat it every time, but this stick is disposable, so once I'm done I'll just get rid of it. Of course I will get rid of it in a special container since this is holding bacteria. This is the culture. I need you just to look at the foggy material on the surface of the culture. This is the bacteria, okay? So here's the thing, this is how I will hold it. I will take some, close it. Now there is bacteria over here. I will apply it on the drop and try to mix it. Make sure to put all the sample that I collected and then I will apply the slide into a hot plate. I kept the ending over here to make it easier for me to hold. Now, as I said, this step is very important to adhere the sample on the slide for fixation, to make the sample harmless, and to adhere it so that when we fix it, so that when we stain it, it won't get off the slide, okay? So I will keep it for like 10 minutes until it's really dried, and then I will start the staining method. Now, as you can see, can you see it over here? I need just to use white background. I don't know whether you can see, it's dried, really dried. Now the reason we did this step, very simple. As I said, we are fixing the sample, fixation, to make this the organism harmless so that we can work with it, and to adhere the sample on the slide while we are staining the bacteria. Now, as I said, gram stain, it comes from a scientist's name. He called himself as a Gram. He discovered the staining method. It's a way to differentiate between gram positive and gram negative. First of all, to view the shape of bacteria. The staining method depends on the cell wall of the bacteria. Now, when you studied in 103 and 101, you know that the cell wall of the organism or the cells has proteins, has lipids. We have or carbohydrates. In bacteria, there is something called peptidoglycan. It's a kind of lipid and protein together. If it's thick cell wall, thick peptidoglycan, this is a gram-positive, and it will be stained by crystal violet over here, and it will look under the microscope. The color of the bacteria becomes violet. Okay? If actually it's stained pink color, it means there is a very thin layer of a peptidoglycan, which in this case, when I do the decolorization, crystal violet will be removed, and then it will be colored with the safranin. Now, we have different steps. First, we apply gram violet, okay? If the gram-positive, which has a thick peptidoglycan, will be stained with the gram violet, the stain will go inside and will be retained. It won't come out when we do the decolorization, when we add alcohol and iodine. This is the second step. And then last step, we add safranin. So it's a violet. It will not be stained with the safranin. It will be only stained with the violet color. So to begin with, we add 1 drop, or to cover the sample, with a gram-positive, okay? And we keep it for 1 minute. So if it's a gram, the bacteria, it's a gram-positive, meaning it has a thick peptidoglycan. The stain will enter the cells of the bacteria and will never come out, okay? So after 1 minute, we will wash it with the tap water, okay? You can see my face over here, that's fine. I need you to see the regular tap water. This is a regular tap water. Look at the slide. The color is violet. Now I will decolorize that by adding grams iodine for 1 minute. Also, I will cover it. I will wait for 1 minute, and then I will decolorize it by applying ethanol over here. I will get rid of all the crystal violet coloration, and then I will apply safranin. So as I said, if it's a gram-positive, if the thick walls are there, the peptidoglycan is thick, the cell wall of the bacteria, even when I do the decolorizing part, the crystal violet will remain, the color of crystal violet will remain inside the cells, and it won't get off. It will be retained inside. So I will again, excuse me, I know it's a pour somehow, but I'm trying to cope. We need to move in the lab. This is why it cannot be done online. Okay, so I wash it for 30 seconds in a tap water, and then I will add decolorizing, wash it, and then I will add the safranin. I think this is a better angle. So in this step, if the gram is negative, it won't be affected with the gram violet. It will be colored pink color. So after 1 minute, I will wash it with tap water. And with a filter paper, this is not a tissue or paper, this is a filter paper, this is specialized paper. I will tap, just tap, gentle tap. I won't rub it, so I don't rub the sample, and I will keep it in the air to be dried and then view under the microscope. Now, usually in the lab we did two kind of bacteria. Let me show you over here. We did Escherichia coli, or we call it E. coli, and we have Staphylococcus aureus. Now, Escherichia coli is a bacteria. Staphylococcus aureus is a bacteria. We will differentiate between them by doing the gram stain, and we will view it under the microscope. Gram stain will allow me to view bacteria, to view the shape of bacteria, as well as to differentiate whether this is a gram positive or negative. Positive is a violet color or purple. Negative is pink color bacteria. Okay, now I will show you the result.